Okay, so we start off with interphase. Before all this, we have uh, a a haploid gamete from mom and a haploid gamete from dad in the form of an egg and a sperm. Uh, they come together to make a fertilized egg, which is a diploid zygote. And once we have that, it goes through interphase and it copies the chromosomes from mom and dad to move on to uh, meiosis 1, where we have two um, two homologous pairs of chromosomes from mom and dad, and we have the centrioles that form the spindle fibers and with the microtubules. But in prophase 1, the homologous pairs uh, do a process called synapsis, where they bind together and they kind of cross over each other, and during that crossing over, they switch parts of themselves, and that's called genetic combination. So, and the homologous pairs do that so that each like daughter cell is different, and then that creates like difference in humans. That's why you're not the exact same as your siblings. Is because these it's because these homologous pairs of chromosomes switch parts of each other. And then we go into metaphase 1 after synapsis and crossing over, and we get tetrads, which are the homologous pairs once they're binded up. And the centrioles are producing the spindle fibers to line them up in the middle in metaphase 1. And then we go to anaphase, where they start being spread apart. So these are still chromosomes. They were just being separated from the homologous pairs. And you can see in here from the genetic recombination, there's they have different swapped parts. So that no two daughter cells are the same. And then we go into telophase, where they start separating and like here, the spindle fibers from the centrioles are pulling the homologous pairs apart. Sorry, I didn't say that earlier, but after telophase 1, it goes to the cytokinesis, and basically that whole process, what we just did, is the same as mitosis, except for the homologous pairs. They just have single chromosomes in mitosis. But meiosis is basically mitosis twice, and we'll see that once we get to meiosis 2 in prophase 2, when we have two daughter cells that have two chromosomes instead of the two homologous pairs that we had at the beginning of meiosis 1. And from prophase 1, the centrioles start to produce spindle fibers again. And then we go into metaphase 1, where they start lining up the chromosomes in the middle with the spindle fibers from the centrioles and both of them. And then after metaphase 2, we go to anaphase 2, where the sister chromatids start being separated by the spindle fibers. And this is kind of the really big difference from mitosis, is that the process happens twice. Meiosis 2 is more similar to mitosis than meiosis 1 is, because meiosis 1 has the homologous pairs instead of the sister chromatids. And in meiosis 1, you're separating the homologous pairs, not the sister chromatids. In meiosis 2, you're separating the sister chromatids. And that's what's happening in anaphase. The sister chromatids are being separated just like in mitosis. Then we go into telophase, when they the two daughter cells we have start to divide, and the cytoplasm starts to divide in telophase. And then we go into cytokinesis 2 when we have four new daughter cells. Okay, so as I was saying, in cytokinesis 2, we form four daughter cells that are all different from the first original cell and the first original diploid zygote from the um, two haploid gametes from mom and dad, and all in the two homologous pairs were combined in genetic recombination. So our four daughter cells all have different amounts of the original chromosomes, and that's how we get different, like that's how we get different people. That's why we don't look the same as our siblings, and this whole process is very important to in human life for reproduction, and so we all can reproduce and have different offspring. They can evolve and adapt to new situations. Oh, and this whole process is mainly like mitosis twice, like I said earlier. We have meiosis one, which is more different than mitosis because we're separating homologous pairs instead of sister chromatids. So meiosis one is just separating homologous pairs, and then meiosis two is separating sister chromatids, just like mitosis. So meiosis two is more like mitosis, but the whole process is like mitosis twice with a little different at the start. So basically, that's it.